Okay, so let's look at innervation of a muscle, and then from that, look at muscle contraction. So nerve impulses cause skeletal muscles to contract. So let's take a look at what is known as a motor unit. Now, a motor unit is one motor neuron and all of its associated muscle fibers. Okay, so it can go, so a single motor neuron, so this is a nerve cell, can go to anywhere from 3 to 1,000 muscle fibers or muscle cells within a muscle. All right, so here our impulse starts in our central nervous system, or brain or spinal cord, and then it goes to a motor unit, which is a motor neuron and the muscle cells it connects to. And here the connection occurs at what is known as a neuromuscular junction. So these things here, these are neuromuscular junctions. This is a blow up of that. This is a region where a motor neuron meets a muscle fiber. Now, the motor neuron, so the nerve cell, and the muscle cell are not directly connected. They have this space in between there. And this is called the synaptic cleft. And so what has to happen here is that we're going to release a neurotransmitter into that synaptic cleft. And that neurotransmitter is going to cross that cleft, and it's going to bind to the muscle cell and then cause an impulse. Now, in the next chapter, we'll talk about what an impulse is, but for this chapter, we're just going to call it an impulse and work with that. The neurotransmitter that stimulates muscle cells to contract is called acetylcholine. This is an abbreviation for it, ACH. So that nerve cell is going to release acetylcholine. Acetylcholine is going to cross that synaptic cleft, and it's going to cause an impulse or an action potential on the next cell, all right, on that muscle cell. Okay, so what's going to happen here is that impulse moves along the sarcolemma, and then it's going to move down the T-tubules. And when that impulse moves down the T-tubules, as I mentioned before, the sarcoplasmic reticulum is found on either side of those T-tubules. So when that impulse moves down a T-tubule, it's going to stimulate that sarcoplasmic reticulum to release its calcium ions into the sarcoplasm. So it's going to stimulate, when that impulse moves down the T-tubules, it's going to stimulate the sarcoplasmic reticulum to release calcium ions into the muscle cell's cytoplasm. So what's going to happen next here is, we can see here, calcium binds to troponin, and that's going to change the troponin-tropomyosin complex shape. Okay, so if we look at this picture here, this is before the calcium binds, this is after the calcium binds. So here, calcium binds to troponin. That's going to change the troponin-tropomyosin complex shape. So this then opens up those active sites on the actin, and that allows myosin to bind to that actin. Okay, so calcium binds to troponin, changes the troponin-tropomyosin complex shape. That opens up the actin. The active site on the actin, that allows myosin to bind. Okay, so what's going to happen next here is that myosin, now using ATP, it's going to pull on that actin myofilament. Okay, so here we have, here's our actin myofilaments, here's the myosin, myosin grabs, pulls on it, that's going to get those these Z discs to come closer together. And so when those Z discs come closer together, that is a muscle contraction. Okay? Now, for relaxation to occur, what we have to have is that nerve impulse stops here. So there's no secretion of acetylcholine in the synaptic cleft. We actually have enzymes in the synaptic cleft that break down the acetylcholine. All right? And so since that, there's no impulse moving across the sarcolemma, no impulse down the T-tubules. And because of that, now we pump the calcium ions back into the sarcoplasmic reticulum. And so here, we're going to go from this back to this. So if the calcium is being pumped out, right, and now calcium is not bound to troponin, now the troponin-tropomyosin can move back into their original position by blocking the myosin from binding to the actin. And then the actin and myosin, they slide back into their original positions. Now, one of the things I left out of here was, and this picture doesn't show it, but there's like these spring-like proteins here that when we get those Z discs closer together, it compresses the spring protein, and then when the myosin releases the actin, that spring protein pushes them back to the original positions. Okay, so that's relaxation. All right, now you've heard of rigor mortis. Rigor mortis is where the muscles stay contracted after death. Well, after death, there's no energy to pump calcium ions back into the sarcoplasm. So what's going to happen here is calcium ions are going to diffuse. So after we die, calcium ions are going to diffuse out of the sarcoplasmic reticulum. They're going to bind to troponin, all right, changing the troponin-tropomyosin complex shape. That allows the myosin to grab onto the actin, and it has enough energy to pull it once. And then the myosin stays bound to the actin then, okay? And so that's what rigor mortis is.